it's good to be here and see all of you, know people that I've heard about, but I didn't have the opportunity to meet. And thank you. As I said earlier, it's good to make connections and see what other people are doing, and make this connection is very important. So thank you for this opportunity. Okay, I'll share my presentation. Hopefully it's gonna work. I think it did, right?
We can see it. Yeah, great.
Great. Well, Patricia invited me to talk about what I have done for a few years. Then we'll discuss about the biological control of ticks with entomopathogenic fungi. So we'll talk about what we have done and about our perspectives of future studies and challenges. So tick are parasites very well adapted to the Brazilian climate. It has a high biotic potential. So one of these females right here, each one lay about 3,000 eggs. So it's a fabric, it's a factory. Sorry, and the conditions in Brazil, they are very promising for this tick biology. So the control of this tick is complex. It's challenging. It's not simple. The chemicals are the most method used to control ticks. That's the method mostly used, and of course it's important to get other to have other methods to join the chemicals, and then we can control this tick more efficiently. If we I used to think about this, how progressive have the studies been with ticks and entomopathogenic fungi for biological control. So when we make a quick search using this Scopus database, we see that the first studies with biological control of ticks, they appear in 1995. This is Vanya's publication. And of course, we have few studies before that, but they are studies not focused on the control. They are like isolation of fungus from naturally infected ticks and this kind of study. But the first one that we have known is the study that Vanya started investigating how effective this fungus could be for tick control. But since 1995, we have about 250 publications. Of course, regarding the use of fungi for tick control or for biological control, we have a lot more. But when we search for studies that involve ticks and entomopathogenic fungi, this number is quite low, 250 publications. And when we compare the numbers with the studies that investigated entomopathogenic fungi for agriculture insects, we see this number is very higher. The publication started in 1967, and we have about 3,500 publications. So there we see how the biological control of insect pests, they started earlier, and they have more people investigating this subject. It's interesting that when we compare the number of studies by countries, we see that Brazil is the country with most expressive number of publications in biological control of ticks with entomopathogenic fungi. This is was quite impressive. But we see that besides that, we do not have a fungal product registered for tick control in Brazil. We do not have this product yet. We do have like MetaHeal, that is a product commercialized by Copart in Brazil. It's claimed to control ticks, but it's not registered against ticks registered for tick control. We keep asking like why we have many publications and we still do not have a product for tick control, and then why we have too many publications on this subject. And maybe because in Brazil, Brazil has the major commercial herd of cattle in the world. We used to say that we have more cattle than human beings in our country. So this is an important reason for the number of publications that we have. Another important reason is that we have identified many populations that are multi-resistant to chemicals. And we see here in some parts of the country, we see populations that are resistant to 8 of the 9 classes of chemicals that we have available in the market. At this time this paper was published, they were all the classes that we have. The 8 classes were all we have, but we have a recent molecule, so that we do not have studies yet, and probably we do not have resistant populations. But the states that we see in white here is not because we don't have any resistant tick population. It's because we have not investigated yet. I know the studies, they are moving on, and they have this information updated, but it's not still published. So it shows us that we do need methods alternative to chemicals to control ticks. And the question is why in Brazil, we have effective programs for biological control of insect pests, and they are very well established in Brazil. They have like huge programs, like millions of hectares treated with entomopathogenic fungi for agricultural pests, and they are very effective, but we do not have a program for tick control. And one reason for that is that we change our target. We have a different target, and a target with a very complex biology and a complex ecology, and that makes this target makes the difference in the program for tick control. And also, as I mentioned in the first slides, the number of people, of researchers investigating this subject that are dedicated to control ticks with entomopathogenic fungi. Well, when we talk about ticks, and we say ticks as they are equal, like as we have only one species, and we know that we have different species with different biology and different behavior, and they are a lot different, and it shows us they are different challenges also. So here we have the cattle tick, the dog tick, and then the horse tick, and here I used to say the everyone tick because Amblyomma sculptum is not that selective. And we see here that here we have one host tick, that it means it depends on of one host to complete their cycle, and then whoops. And we have a 3 host ticks that they need 3 hosts to complete their cycle. So they have different behavior and different biology. I think it's important to mention about the life cycle. When the larvae reach here we're talking about Hypcephalus micropolis, that's the cattle tick. When the larva is in the pasture and then we reach when they reach the calf, the larvae start sucking blood, and then they molt to nymphs and then to adults, and then the females, they get engorged of blood, full of blood, and then they drop off the host and lay their eggs in the pastures. Okay, and they lay those towns of eggs here, and then they die. And here this tick may have several generations during the year. It depends on the climatic conditions, depend on many things, but usually we have about like 3 to 5 generations in a year. But here with this Amblyomma sculptum that needs at least needs 3 hosts to complete their cycle, we see the larvae reach the host, the first host, they suck blood, and then they molt in the pasture, and then the nymph go to the second one, and then they drop off the host, they molt, and then the adults go to the third host, and this cycle takes a year to get completed. So it's a different world if we compare those 2 species. One thing that is interesting to mention is that 95% of the tick population we see on the host and 95% of the ticks are in the pastures, in those females laying eggs and the larva. And then it makes sense that if we look for the biological control, the application of biological control agents, we need to reach the target. So we have studies that are dedicated to apply the fungus in the pastures to control the larvae and the females that are laying their eggs. And then other studies are dedicated to apply the fungus to ticks on the host. We have many species of fungus, and only in this phylum, Ascomycota, we have more than 64,000 fungus. And we have, of course, other species in other phylum that are also able to infect and kill ticks. But here in this phylum, the most investigated fungus, or the most investigated, the species in these 2 complexes are the most investigated fungi for tick control. So Bovaria bassiana and Marrhesian anisopoli. We have other fungal species that kill. They are very promising for tick control, but these 2 complexes are the most investigated. The infection, the fungal infection is about the same as insects. Okay, so the propagule, they reach the cuticle, and they start germinating. They produce enzymes and penetrate the cuticle, reach the hemocell, and start producing other cells like we call hyphal bodies, and then they colonize the whole tick, and then they start producing the new propagules to spread in the environment, and it goes to the cuticle again to produce these propagules, and that's why we see the tick in green, as we see here, if it's infected with merahizin, or in white if it's infected with Beauveria, for example. And we see here that the cuticle is the first barrier. So the fungus need to penetrate the cuticle to infect the tick. And we notice that different species, different tick species, they have different sensibility to fungal infection. And then we investigated here if these species that are important in Brazil, if these 4 species, if they have different, if the cuticle represent a different barrier to the fungus. And we compared the chemical composition of these cuticles, and we investigated the hydrocarbons of these cuticle. We extract the cuticular lipids from these ticks, and we see that they are different. So, and we start expecting that if these chemicals could limit the infection, the fungal infection. And then we extracted these lipids. I mean, before that, we investigated the germination of these propagules on the cuticle, and there's different species. And we see here that in merahizin and Beauveria, in the cuticle of Amblyomma sculptum, we see here a delayed germination if we compare with the cuticles of other ticks. So it spent more time to germinate here, even merahizin robertsi or Beauveria bassiana. And then we investigated using cytometry. We investigated if what happens if the conidia is exposed to these cuticular lipids. And we see here that the cuticular lipids of Amblyomma sculptum and Dermacentor nitens, they cause conidial death to both merahizin and Beauveria. We don't see these effect, or very small effect against small effect of the lipids extracted from the dog tick or the cattle tick. But these effects are very expressive with these 2 species. Of course, ticks, they have different mechanisms to defend themselves from infection. And when the tick got infected, we also have the cellular immune system, the humoral immune system that defend the tick from this infection. Most of that, what we know about these mechanisms, we import from the knowledge of the defense mechanisms of insects. We still have a lot to learn, and Patricia is the one that is dedicating her research group to investigate a lot of this. So we still have a lot to investigate. I have some students that are investigating this, in fact, but we still have a lot to learn. We have many, in this infection process, we have many directions, because the infection process is very complex. In this paper, we investigated the fact of the transcription factor MSN2 to the pathogenicity of Beauveria bassiana. So we want to know the impact of this transcription factor to the infection. And we made an assay and exposed ticks to the wild type Beauveria bassiana and to the mutant that has or doesn't have the the transcription factor gets deleted. And we see here different in the percent control. The mutant one has a lower percent control. Of course, it's not zero or extremely different from the wild type, and that's what we were expecting, because as we said, there are many factors involved in this infection process. We are deleting one single gene, okay, or one part of this process, and then we investigated why it happens, and we checked the germination of the fungus on the cuticle, and we didn't see difference in numbers. So the wild type or mutants, they germinated about the same, and it was expected because they all got infected. Okay. Then we checked also the quality of these germination tubes, and we didn't see difference either. So we moved one step forward, and we investigated the penetration of this fungus to the tick cuticle, and we see here the wild type, the penetration seems okay, the fungus penetrating through the cuticle, but with the mutant one, we see kind of a limiting penetration, or a delayed penetration at least. So it means the fungus may penetrate because we see infection, but here we see it's 120 hours after treatment, and we see a delay here. And then we check what's the reason for that, and then we notice that the protease activity of this mutant strain is very low in comparison to the wild type strain. So that's possibly one of the reasons. It's interesting that most of studies investigate the use of entomopathogenic fungi against ticks, they use aerial conidia. But we know that the fungi, they have many types of fungal propagules. So we have aerial conidia, of course, but they also have blastospores that are analogous to the hyphal bodies that are inside the host. But we also have submerged conidia, microsclerotia, and chlamydospores. We have other fungal propagules that are not very well investigated, and motivated by some papers that were published, we investigated the effect of blastospores to cause infection in Hypcephalus microplus, and we see that the blastospores were able to germinate on the tick cuticle, were able to penetrate through the cuticle, cause infection, and sporulate in the tick cadaver. We also noticed that the blastospores were able to penetrate through natural openings, and we see it depends on the isolate, but we noticed that some isolates we could see that blastospores were more effective than their conidia. One thing that the literature indicated is that blastospores would be a lot more sensitive to heat or UV, and this could be one of the reasons for these propagules had not been investigated. They didn't have attention, and then we are trying to discover, well, how sensitive they are. And then we investigate the sensitive and compare the sensitivity of blastospores to the conidia of the same isolate. And we notice here that in most cases, blastospores are in fact more sensitive to heat, but we also see isolates where both conidia and blastospore are equally sensitive to heat. But we notice that in some isolates, blastospores could resist high temperature for a considerable period of time, and that could be enough to the fungus to cause infection. That is the interesting thing. The same thing with UV radiation. In most of isolates that we investigate, they are in fact more sensitive, but they are not too sensitive. I mean, maybe they are enough tolerant to cause infection. And then we know that they penetrate faster than the conidia, and then maybe they are less exposed to environmental stress than the conidia. That takes more time to germinate and penetrate the cuticle. We test one blastospores in the field, spraying blastospores over naturally infested cattle. It was a very challenging test. Valeska here in this picture this test was part of her PhD. It was very challenging study because we have to mass produce blastospores. We have to formulate this product to apply on the calf and evaluate the efficacy of this study. It was very challenging for us. This was our first field study. The results that we got, we test the same isolate we test in the field, and we see the same response here. I mean, blastospores were more effective than the conidia, but the numbers were quite low. We were expecting, and we could see the higher efficacy number we got was right after the treatment. It was in day 0, and then the numbers start, the efficacy start going down. We know that we still have a lot to learn in this assay. We are still investigating other mechanisms and different formulations that could be more effective for tick control. We are also investigating the effect of microsclerotia. That's a different propagule. This is not infective. It's not an infective propagule, but we formulated this propagule in the field and make those beautiful granules here. And if they're not infective, so how do they act? How do we use them? Well, these granules, they are not infective, but then when they are exposed to optimum conditions, high temperature, optimum temperature and high humidity, they start sporulating, and then they produce the conidia that are infective against ticks and other arthropods. And we test this in a Petri plate. We applied these granules in a Petri plate and let them incubated for a few days to sporulate, and then we put one engorged female here, and this female, they get infected and they die before they lay all the eggs they could. And then we see here, we test different isolates, and we see that some isolates were more promising than the others, but we could see that some isolates were effective in reducing the number of eggs. We count the number of larvae, okay? So we see that they reduce the number of larvae they could generate. Then we did other tests when we exposed the fungus to different temperatures. We exposed to 32 degrees. It could be challenging for the fungus, challenging also for the tick, but we still could see some good results. The microsclerotia were able to produce infective conidia even under 32 degrees. Then we move one step forward from the petri dish to semi-field conditions, and we apply those granules on the soil, and then we count the number of larvae, and then we see we could see good results. We're still trying to understand and how to make this more effective, but we could see significant difference in some of the treatments that we had. One step forward more, and we are trying this in a large scale, applying this in the field. And we did this during the rainy season, and we could see that the groups treated with the fungus, they have a good efficacy in the second week and the third week, and then the fourth week on, it was about the same, but we have a 54% efficacy. Studies in the field are challenging because we see those differences in temperature. Here we had about the mean temperature 23 degrees and relative humidity about 86%, but the maximum temperature almost reached 30 degrees and the minimum 19 degrees, and here was the precipitation. And this is different because how is this precipitation? Sometimes we have like a very hard and strong rain during the night, and then it's hard to understand how these effects will interfere in our results. But the results were interesting, 54% efficacy. We do have challenges to face. We had a varied efficacy in different studies. If you see here, we see 50% efficacy. We could do the same experiment in a different period, and it will have a stronger rain, or you have 1 or 2 days that were very hot, and these variety of results are very sometimes are complicated to explain. We also need adequate formulation of propagules, and we have to think on our target. Where is our target? So it needs an adequate formulation. It's different to prepare formulation to apply in the pasture than apply on the host, right? So we need adequate formulation, or it depends if you're formulating blastospores or if you're formulating microsclerotia or conidia. They do need different attention for this the preparation of this formulation. Registration rules are also challenging because biological agents are still faced as chemicals, and we need different rules because it's a completely different thing. We cannot put the biological control in the same box that we put the chemicals. So they are different proposals. So they have different manner of action, and it's a different world. We cannot put them in the same rules. We need strategies for publication, for application of biological control fungi, and maybe they change depending on the species, and and some of these strategies are focused on application when the tick infestation is not too high. Okay, because in this case you need a very rapid method to control, and this may cause some economic challenges because you have to convince people to treat cattle when it's not a problem yet. Well, I'm 5 minutes over my time, Patricia, but I think I hope I have covered all the subject I was expecting to talk. This is my team that I'm very proud of them, because they are the ones that produce all these data that I brought here. And this is our university. This is our collaborators. And Patricia, Isabelle, and Vanya are very close collaborators. And this is us in the Instagram, so you want to follow our works. Thank you very much. Thank you again for the invitation, Fitoini and Patricia. I'm very glad of being here. Thank you.